Hello, everyone. I'm extremely happy to be here to share my latest research on building AI models that can enable new biomarker for diagnosing and assessing Parkinson's disease. Okay, so to motivate things, let me start with some famous people you might have heard about. Muhammad Ali, the most famous and beloved boxer. Michael J. Fox, one of the most well-known TV show and movie star. Brian Grant, a famous professional basketball player at NBA, and Michael and Ozzy, who are one of the most talented people in their field as NASA astronaut and rock star, respectively. So what do they have in common? Of course, they're all famous and probably rich. But what I'm going to tell you is they're all diagnosed with Parkinson's disease. And of course, they're just a very small portion of the people who have Parkinson's disease, or PD in short. So what is PD? PD is the fastest growing neurological disease in the world and the second most common neurological disorder after Alzheimer's disease. Over 1 million people in the United States and 10 million people worldwide are living with PD as of 2020. Which results in an economic burden of $52 billion per year. Unfortunately, so far, no drugs can reverse or stop the progression of PD. As a progressive disorder, Parkinson's disease often begins with subtle symptoms such as slight hand tremor. It also affects the nerve system and eventually leads to uncontrollable movements, shaking, stiffness while walking, and balance issues. And over time, people's speech can become slurred and their facial expressions fade away. A key difficulty in PD drug development and disease management is the lack of effective diagnostic biomarkers. The disease is typically diagnosed based on clinical symptoms related mainly to the motor functions, such as tremor and rigidity. However, motor symptoms tend to appear several years after the disease onset, which leads to late diagnosis. Often by the time when motor symptoms of the disease first emerge and a diagnosis is made, such as the highlighted region in this picture, the disease has already been developed for tens of years, and a large percentage of the dopamine-producing neurons targeted by disease have already died off. Therefore, there is a very strong need for new diagnostic biomarkers, particularly ones that can detect the disease at an early stage. Additionally, over the years, researchers have studied cerebrospinal fluid, neuroimaging test, and blood biochemical test as potential method to detect Parkinson's disease. However, such approaches are pretty invasive, expensive, and require access to specialized medical centers. This would hamper the regular testing that can lead to an early disease diagnosis or continuous monitoring of its progression. Also, in addition to just diagnosing PD, there is also a strong need to determine the severity of PD, as well as tracking its progression over time. Tracking the status and severity of the disease is important, as in different stages, clinicians will use different medication and therapies for personalized treatment. However, today, the assessment of PD progression relies on patient self-reporting or qualitative rating by a clinician. Typically, clinicians use a questionnaire called MDS-UPDRS to assess the disease severity. This MDS-UPDRS is semi-subjective and does not have enough sensitivity to capture small changes in patient status. And as a result, PD clinical trials often need to last several years before the changes in MDS-UPDRS can be reported with sufficient statistical confidence, which increases cost and delays progress. Therefore, Effective progression biomarkers for tracking the severity of the disease over time is also of great demand. Okay, so far we know that there are two key challenges in PD. The first one is how to diagnose it effectively and also in an early stage. The second one is how to quantify the severity in an objective and easy way that can also be repeatedly measured over time. Fortunately, there are already some evidence showing that non-motor symptoms might be indicative of early PD diagnosis. A relationship between PD and breathing was noted as early as 1870 in the first work when PD was observed. Over the years, this link was further strengthened in later work, including works that reported degeneration in areas in the brainstem that control breathing, and weakness of respiratory muscle functions, as well as the sleep breathing disorders in PD. And interestingly, clinicians have observed that these respiratory symptoms often manifest years before the actual clinical motor symptoms, which indicates that breathing and sleep could be promising for risk assessment before clinical diagnosis. So motivated by this evidence, in this work, we seek to use machine learning or AI to develop such a diagnosis and progression biomarker for PD using nocturnal breathing signals. And when talking about machine learning, we have all seen the tremendous progress of machine learning recently, from things like GPT-3, which uses deep learning to produce human-like tests, and machine learning translation systems, which can automatically translate across different languages, and to models that can classify images extremely well and even better than human, and to systems that can generate realistic images just from text. So to design our biomarker, since human breathing and sleep are in nature complex and high-dimensional, we seek to design or use AI-based system for PD diagnosis and assessment. Specifically, our AI model first extracts nocturnal breathing signals either from a breathing belt worn by the subject or from radio signals that bounce off their body when asleep. By either of these two procedures, we obtain a full night of breathing signals from the subject of interest. It then processes the breathing signals using a customized neural network and infer whether the person has PD, and if they do, assess the severity of their PD in accordance with the MDS UPDRS. So when we want to integrate the system for the real-world deployment and data collection, we are actually using two main methods, which as shown on the left, we are using a breathing belt to collect the breathing signal overnight. And we can also ask the participant to install a wireless device in their own home, as shown on the right figure, for sensing the nocturnal breathing in a touchless manner. So for the second scenario, when we are sensing the breathing using wireless signals, past works already show that we can monitor humans breathing remotely without any physical contact. And as shown on the right, by transmitting wireless signals and analyzing the signal that bounce off the body, we can measure the breathing accurately, as shown on the left. Here, for the below part representing the inhale of humans breathing, and the upper part representing the exhale of the human breathing. And the amplitude of each peak represents the volume of each breath. So with our AI model and customized algorithms, we can actually achieve very good performance for both data types, like the breathing belt or wireless signals, with an average accuracy of 90%. And moreover, since for each individual, we can collect multiple nights of data, and by combining the predictions for multiple nights, we can even achieve a very good 100 diagnosis accuracy over both control and PD subjects that have multiple nights available in our study. We also test the model performance when the testing distribution is different from the training. Namely, we want to generalize to the unseen population or institutions. The result shows that even when testing on an unseen institution's data, the performance is still good with an average of 85% accuracy. And our system is also able to detect the severity of PD with a high accuracy. And compared to two gold standard metrics in the PD field, one is the continuous MDS-UPDR score, and another is the discrete H and Y stage. Our model prediction is highly correlated with both gold standards. And one interesting observation is that the model also shows initial evidence for early diagnosis. In our dataset, we identify a group of patients who were diagnosed with PD later, but were not diagnosed 6 years before. And even though they were not diagnosed, they're likely to already have PD onset at that time. So we directly test our model on this data and found that the model is able to distinguish this high-risk prodromal PD group with age and gender matched controls and with a high statistical significance. Moreover, since our solution can measure the breathing every day, we can even track the disease progression in a very fine-grained manner. Here, the yellow bar is the gold standard PD questionnaire, which is done 3 times a year, and with potentially very high variance. In contrast, our model can provide a robust and more frequent assessment at home, which is desired and more meaningful. I also want to highlight that the work has also the potential to speed up drug development with the newly found digital biomarkers for both diagnostics and tracking progression. And using such an AI model that is capable of detecting subtle patient changes and responses to new therapeutics, we can have the potential to accelerate clinical trials and reduce costs and inform more effective treatments. So to conclude my talk, we have proposed a new AI-driven health application that detects and assess Parkinson's disease using machine learning. And looking beyond, I believe we still need more robust and generalizable learning algorithms that can work in the wild to support more real-world healthcare applications. And thank you for listening.